Syracuse University in the physics department, and she's doing some really great stuff trying to understand how cells move around in space. And she either looks at mammalian cell culture or bacterial cells and utilizes some really cool three-dimensional ways to control that space that they move around in. And we also have Kathy High from RPI, which Mariana will tell you about. But I'm really excited about her talk too. You're a great scout.
Hi. She's an artist and a bio artist. That's why this is the reason to invite her here, but also because she's an amazing artist in general. And she's not only a bio artist. A lot of other things, an amazing person, and also one of my favorite professors because I studied with her at Rensselaer, and I just wanted to let you know about a few things that we did together related to some kind of a fuse between biology and art, and I think this is another reason, not only the art that she's going to present that traveled around the world and was exhibited not only in the United States but in Europe, in Australia, and like all over the world. But I just wanted to say something which is in relation with our initiative here, which is the BioArt Mixer that we started with Heidi a couple of years ago. When I was a student at RPI, Kathy was the professor that initiated this thing which we call BioArt Initiative. And it was a long-term, relatively long-term, of collaboration between the life sciences and the art department, which manifested itself in exhibitions. We expect to have an exhibition here, a bioart show in a couple of years as well, and also bioart mixers. So they kind of borrowed this kind of a name, which were actually really great events. And now I think these whole ideas of the fuse of arts and sciences, it expanded towards Sanctuary for Independent Media, which is culturally, it's a community involvement center, and a lot of programs for young people who go to Hudson and see what floats in the water, or what like these kind of things. So really engaging programs for people from diverse backgrounds and diverse ages. So I hope Kathy will tell us maybe in the Q&A or maybe in the presentation also about this. So
thank you.
Yeah, that's right.
Okay. First comes science.
Hi guys. I'm really excited to be here. Thanks to Heidi and Ed for the invite. It's a little different than what I usually do, so if anything's unclear, just shout out your questions. I'm happy to take them. So I moved here a year ago. I came from Philadelphia, and I'm starting out my lab here in the physics department. And we kind of have two sides to the lab, either looking at animal cells, which you see here on the left, and on the right is a culture with bacteria. And a lot of my colleagues in the physics department use telescopes and like to look at astronomy, but I really get my inspiration when I'm looking down in the microscope and see these, beautiful pictures and things at the microscale that inspire me. So I thought I would share some of that with you. Actually, some of my favorite work that I found the most fascinating. And this started actually with some interests I developed a few years ago now, thinking about this problem of collective motion. And if we open our eyes and look around, we'll actually see that this occurs all around us and at many scales. So actually right now we're herding into a room. Okay, there's some people moving together. But we see there's flocks of birds, there's beautiful schools of fishes. And if I look under the microscope, I see that bacteria, which are millions of times smaller, also are displaying collective motions, actually very dynamic motions that I'm going to be showing you. So these things are very different. They have very different interactions. Colonies of individual bacteria don't have brains like we do, but they still exhibit these collective scales. And I'll show you another example where you may have seen this. Maybe you've been involved in a mosh pit, which is actually one of the model systems for this collective motion. And you get swirling patterns that develop. Actually, this was work done at Cornell, which is obviously very close by. But let's go back and think about how a microbe moves. So I'm going to be telling you about Serratia, which is very closely related to this famous organism called E. coli. And E. coli have flagella in their cell body. And these flagella have a little motor. I don't have a laser pointer. But here, the motor is connected into the cell membrane. This is an artistic description of all the molecules in the cell. And this little thing, which is only 20 nanometers, actually rotates. And that's how these little guys move around in a fluid. And we can visualize this. This is some work from Howard Berg's group, one of the first groups to show this. They have the cell fluorescently labeled, so you can see the cell body and the flagella, and they're propelling forward.
Is it random, or is it programmed, the flagella?
Is it random? So they go in a certain direction for a while, and then they reorient. So at a very simple scale, over a very long time, it becomes random. However, oftentimes they will sense a nutrient, and they'll want to go towards that nutrient, and then it's no longer random. Yeah.
By the way, that's exemplary of how we want this audience to behave. Thank you, David. Shout out questions as they come up.
All right. Okay, so I'm going to show you some videos. Oh, I was rearranging my slides and I took it out, but I promise I'll show it to you later, of this turbulent behavior in the bacteria when you put them very close together. You get these turbulent flow patterns that are pictured on the right. And I just want to highlight that this is reminiscent of perhaps another very beautiful problem you've seen in physics and fluid dynamics, which is the question of turbulence. So this is actually a sketch from Leonardo da Vinci. So I thought I would put it side by side here. So turbulence is actually a very old problem, hasn't been solved. Trying to understand the flow of vortical structures that you get. But what I can tell you, even if we haven't figured it out, is that's not what the bacteria are doing, okay? What we know about fluids at these large scales is that you need a lot of inertia, you need a lot of momentum, which you would see if you pour out a bucket of water. The bacteria that are so small, they just can't generate that inertia and momentum, which would lead to these vortical flows. So we've been trying to investigate how do these flows emerge, why do bacteria do that, and I'm going to tell you a little bit how we've been looking at that. All right, so this is the video I wanted to show you. So this is actually slowed down. If you look under the microscope, it's going even faster. But I slowed this down so that you can see it. This is what we call a swarm of bacteria. It's Serratia marcescens. Serratia marcescens is the pink scum you get in your bathtub when you don't clean it, okay? So you probably are familiar with them. So they're swarming around here, and you know, as a physicist, we like to play certain tricks to figure out how this material is behaving. And one of them is we like to see how it interacts with something else. Okay, so here I'm imagining that we have a passive phase of matter that's stuck there. So it's just kind of like the bacteria, but it's not moving, it's stationary. And I want to know how this active system would interact with something I can describe as a physicist, so something that's still and stationary. And, well, this is what that looks like. Okay, so this is a video that I've sped up, but I have a passive phase that starts there in the beginning. And you can see there's these vortical flows of bacteria swimming around it. And eventually they're able to take that dead passive regime and take it away, and they're able to capture that new territory. Okay, so let me tell you what we did. A very simple trick. We just killed those bacteria very quickly. We isolated them. We took UV light, and we shone it very localized in this active swarm. So we can actually create different shapes, whatever shapes we want. And we isolate this phase of the material. And what I just showed you, it's catching because it almost looks like it's a melting, like it's melting snow, right? But I can tell you, we can describe how snow melts. It's because there's something called a chemical potential, and chemicals have certain bond energies between each other. But that's not happening here, okay? I can't define things that I would define for a normal material, like viscosity, because this material is already moving without having to apply any force. That's a very unusual property. So I wanted to figure out what's going on, and this is just kind of the schematic. Maybe you've seen in a textbook where you have a solid and a liquid, like maybe you have melting ice, and then compared to this new, what I'm calling a new type of interface, something that's dead and living. So we're at the edge of living and dead. All right, so how does this evolve? So this captures that, that transient behavior, right? So we have this disappearing boundary. And I really wanted to include this figure for you guys because we can track the vortical flows that are going on in these structures. So we have streamlines here plotted on the right at different times, and I've highlighted by color individual vortices. And these vortices in the flow can actually move around, and they can hit that boundary. They can stay on that boundary. They appear, they disappear. It's a very dynamic process. And we don't understand how those vortices form as of now. So how does this work? How do we get melting ice with a bunch of bacteria? So again, this is my other trick I like to play as a physicist, is I like to track individual particle motions. Yes?
How do those vortices interact with each other versus the passive space?
That's a really good question. We haven't tried to answer it. Yeah. It's, I'm sure, very different, but I don't know. We've done a little bit of work, and we found that those vortices, if they're near the boundary, they tend to last longer than if they're out here. Yeah. Okay. So this is the edge of the swarm, and like I said, I like to do little tricks where if I want to understand the flow, if I want to quantify the flow, I'm going to put little tracer particles in there, and I can track where each of these particles go. All right, and I love this video because it's something stationary and then it gets swept away into a flow pattern. And then we can if we track all those particles, we can overlay the trajectory of each of those particles with its own color and see the beautiful dynamics. So this is a problem I'm still going to solve, but haven't yet. But I use this trick to look very close to the interface to find out what was going on at that live dead interface. And what we saw is that here there's the active regime, we got the particles moving on. There were particles that were stuck here in the dead regime. And if you watch long enough, they're still, then they'll start to jiggle just a little bit. See, they're starting to move. Now it gets swept away into the field. So it looks like the active bacteria are, like, through pushing interactions, getting those bacteria to move away. Okay, so I am the scientist tonight, so I'm going to try and explain one figure. Sorry to put you through this. If you're just interested in the art, then you can tune out and come back in the next slide. But we wanted to come up with a quantitative relation to figure out how this boundary was moving. So taking actually a hint from how snowflakes formed, we thought we could relate the interface motion to the curvature of the interface. So if it's sticking out, it's going to erode faster. And we assumed the speed of the swarm, because that was the part pulling the bacteria away. So I have this hypothesis that the velocity of the interface depends, there's three constants there, A, B, and C, and then I have curvature and the swarm speed, which are dependent variables. All right, so I have this equation. Yes, I'm going to make you think about it. I have three dependent variables there. This is an equation of, anybody? Are you smarter than a fifth grader? If I have two variables, I have an equation of a line. If I have three variables, I have an equation of a plane. So what we did was very carefully, we went through all the data, we figured out the curvature of the interface, we figured out the speed that was hitting the interface, and the motion of the interface. And if we plot that data in a three-dimensional coordinate axis, we get data that collapse sort of toward the plate. But of course it's noisy because it's this biological system. So we were excited about this because we had this active material that was very different from passive systems that people are just starting to look at. And we were able to at least figure out some ways about how it moves. All right, let me just say, why does this matter? Why do microbes matter? And it's important for health. I'm just highlighting one example. This is a bacterium that infects catheters, right? This is a little piece of catheter, and you can see that it's crossed over just by going over that catheter. So can we design the properties of that catheter to inhibit bacteria moving into the space? The open question. I really love this problem too. This is mixing of different microbe species. And this is some work that was done with Proteus. And bacteria can somehow sense whether they're similar, whether they're the same strain when they collide, or whether they're different strains. And if they can do that, they'll either form these very sharp boundaries where they don't mix, or if they say, hey, this looks like someone I know, they'll mix. And I think that's a beautiful fluid mechanics problem yet to be solved. And then also these microbes are really smart. So some species they get other bacteria to do the work for them. So some bacteria species aren't motile, they save their energy not to do that, but they'll get pushed around by others. Yes?
In terms of the not mixing, has it been separated from like chemotaxis and just sort of like chemical signaling as opposed to like fluid dynamics?
What is chemotaxis? Chemotaxis.
Chemo. So chemotaxis is when bacteria migrate towards a chemical signal. So that is definitely involved here, and it's all out warfare, like chemical bombs going off to try and kill the other species. I don't think the contribution to the mixing and the physical component has been figured out. Yeah. All right. Let's see, how much time do I have? What am I doing with time? Okay, so I'll just highlight some of the current work we're doing. So this is actually in collaboration with Roy Welch's group. This is Myxococcus xanthus, and it's expanding out on a surface. And the goal that we're trying to do here is to control the mechanical properties of that substrate to either make them swarm out faster or swarm out slower. So some of our preliminary work shows that if we make the surface stiffer, that the bacteria are able to migrate across the surface faster. And I just want to also say, we kind of do this too. So these are mammalian cells. Actually, we got these cells also from Heidi's group. Thanks, Heidi. And mammalian cells, which are very different, they're squishy. Forgive the little glitch there. It's a new lab, still figuring things out. What we can see is that if we look very closely, these cells also have collective flow patterns that they exhibit. And on the right there, this is the velocity field showing collective migrations. And on the right, we have the streamlines that highlight where the collective flows are. All right, so, I'm not really going to give conclusions. I'm just going to give more open questions because that's what why I love science. It just opens up more questions. And so, how do those bacteria vortical flows form anyway? And just, kind of give you the idea that these active materials don't fit into our traditional, passive mechanics ideas of what materials should be. So how do we describe them? What techniques can we use to describe them? And I'll acknowledge some of my group members who contributed to this talk. Some of them are here. Meryl and Max. I didn't see Michael. But, all right, thanks. I'll take any questions.
everybody. It's really lovely to be here. So thank you for inviting me, both Alison and Ed and Boriana. Where'd Boriana go? Oh, there you are. Hi. So I'm going to whisk through a bunch of different projects, and hopefully we'll try and make some kind of resonance between Alison's great talk in some ways, but I'm not sure. At least I work with bacteria sometimes, so there's that. So I am coming from RPI, Rensselaer Polytechnic Institute, which is on the Hudson, as Boriana said, north of New York City, about parallel to here. It's pretty much primarily an engineering and science school, so it's great to be an artist there because, you know, it's very different than teaching in an art school and very challenging sometimes, but wonderful. I love working with the scientists and the artists there. In fact, they've given me a lab in the Biotech Center. I think I'm the mascot. I'm like the art mascot. They don't quite know what to do with me. But this is an advancement from when we had the BioArt initiative that Boriana was a part of literally 10 years ago. We had wanted to get our hands into the lab itself to do research, and now we can do that. So that's the big leap. Here's a picture of the bench. You know, just it's a bench, but to me it's very exciting. I also work at the Sanctuary for Independent Media that Boriana mentioned, which is a community arts organization. And we have all kinds of activities there, including a low-power FM TV station, gardens, and all of these different things. You can go to the website, mediasanctuary.org. I'm the coordinator of Nature Lab, which stands for North Troy Art and Technology and Urban Research in Ecology. It's always my test to see if I remember that. And it's a new project we started, it's not that new, but 5 years or 6 years ago, and we're actually making an environmental education center, but we are very interested in the kind of intersection with this organization of science, media, and art. The three things that are really under attack in our country right now, by the way. You might have remembered that. So it's, we work to bring art. Boriana just came and did a really great workshop with us on, and Alison too, on, Heidi, sorry. You're welcome to come. So, on chimeras, so on genetic modification, which was really wonderful. And so that was really well received by the community. So those are the kinds of things we do there. We are in one block in North Troy, right near the beginning of the Erie Canal. And we have been working to recoup these abandoned lots and abandoned buildings in that block. So we're turning an abandoned building into the new nature lab. We have lots of gardens in the abandoned lots and things like this. So it's a really wonderful place. If you're ever passing through, please feel free to visit. My background's in filmmaking. I was very interested in the medical institution and kind of critiquing the medical institution. This is an older film on new reproductive technologies. Also very interested in relationships with animals, human, non-human relations. So these are sort of things that I was working on when I was working in film, primarily. And then I moved into BioArt when I moved up to RPI, which was in 2002, because I had many more resources moving from New York City. So so I'm just going to talk about these projects that sort of are grounding my work right now. This is a collaboration with a friend of mine, Oliver Kellhammer, who's another artist. And we have been very interested in looking at ruderal ecologies. Does anybody know ruderal ecology, what that means? Yeah, I didn't either. Ruderal ecologies are things that grow out of the ruins. So think of what happened in Europe after the World War II, for example, where things were growing up out of literally these cities that were bombed. And that's how the term was developed, and watching that kind of growth come up. So we're interested in that because North Troy and Troy in general is an industrial city, much like Syracuse is, post-industrial. And there are lots of sort of brown fields and sort of dead areas, but there still is all this rich life. And so what does that mean going forward? Oliver and I worked on this. This is Oliver. And he is a really wonderful person. As an artist, he's great, but also he's a permaculturalist. So he's working with plants all the time and building this into his artwork. But he asked this really great question, which is, are there correlations between what is happening in our bodies, in our guts, and on our skin to the microbiological processes occurring in urban air, soils, and waterways? Are there novel symbiotic partnerships for bioremediation and detoxification evolving under our very noses? So we're sort of looking at these environments, but also thinking through things like what's happening in our bodies. So I'm interested in bioart, as Boreana's told you. I'm going to skip through these slides because you all probably know a lot about bioart by now. No? Okay.
Don't skip too much.
Anybody recognize this image? Are you horrified by it, or is it just sort of like, it's just an ear on a mouse? Okay. So in 1997, this was a horrifying image to people. I remember this. I'm older, really old, so I remember, like, oh, horror. Now people are like, ear on a mouse. There are a lot of artists who've been working with other species. Marta Jimenez is one, and I'm going to tell you some of my favorite people. I would have put Boreana in here, but I'm assuming you've seen her work. So Marta Jimenez is an artist from Portugal, and she did a project early, in like 20 years ago now, where she took a hot needle and stuck it into the cocoons of butterflies to make these extra markings on their wing patterns. And so, and they have no nerve cells in the wings so that they didn't feel any pain when she did this. People ask that question, so I have to say that. But she's asking this question of, what is nature? Like, we always have this nature and culture divide. She's asking, is this still natural? What is the butterfly still natural now that she's sort of involved herself into its design? Another artist who I love is an artist who's a scientist and an artist, Brandon Ballingy. And he's very interested in environmental work and studies sort of toxins in our waterways. So he looks at amphibians and fish and tries to track them in ways so that we can see what toxins have done to their bodies. So this is a print that's about the size that it's shown here, and you can clearly see, because of the way he stained the frog's body, that it's got these mutations of these extra legs. But I find this work really beautiful. He's living in Louisiana. He's been working on the Gulf Coast. And it's really stunning work that he does. And he also is a citizen scientist. He takes people out to do research in their own communities. So these are people I'm kind of model my own practice after. Tissue culture and art are people who live in Symbiotica and live in Western Australia and run this wonderful program, Symbiotica, which is an art and science residency program. They work with tissue culture to make their sculptures. So they're actually sculpting them into shapes like this little jacket here, right? You see the little jacket. So this is called victimless leather. And victimless leather is supposedly, like we aren't using the cow anymore. We're just using the cells of the cow. They always talk about this work ironically, though. First of all, the little jacket can't live outside of this system, this bioreactor system that's sustaining them. And then the other thing is, part of the medium that's feeding the little jacket comes from bovine serum, which is from animals, from calves. And so it's like an ironic system, thinking we're getting out of this conundrum of working with leather and things like that, but it doesn't really do that. So I'm going to go through some animals that I've been working with, transgenic rats, for example. This is a piece I did in 2004 to 2006 called Embracing Animal, where I worked with transgenic rats. And they were in two different exhibitions, but one of them was in Mass MoCA, which is a really wonderful museum if you're ever passing through the western part of Massachusetts. It's in a huge old electronics factory. And so they keep the exhibitions up for like 10 months. So we built this giant penthouse for rats. And I was able to purchase 3 transgenic rats that had been designed to work with autoimmune problems, much like my own. I was interested in sort of looking at the reverse engineering of who do they do the pharmaceutical research on? Whose bodies do they do this on? What are these rats like? And it turns out they work with transgenic rats, rats that have been infused with human DNA, with their own DNA, which is why they're transgenic. And so I was very moved to be, and very naive, and was able to obtain these rats sort of under the table, kind of not. That's a long story. Anyway, I was influenced by this quote by Donna Haraway, Oncomouse TM, which is the cancer mouse. The mice that are bred reliably breed cancer tumors. So Oncomouse TM is my sibling, and more properly male or female, he, she is my sister. Her essence is to be a mammal, a bearer definition of mammary glands, and a site for the operation of transplanted human tumor-producing gene, an oncogene, that reliably produces breast cancer. Above all, Oncomouse TM is the first patented animal in the world. By definition, then, in practices of material refiguration, she is an invention. So this is what these rats are as well. They're bred to be sick. But we cared for them with homeopathy, alternative medicine. They saw a vet every month or every 2 weeks if needed. They were fed alternative foods and also had lots of areas to play. And it was a successful, show. But the big success for me was that the night watchman fell in love with them because we didn't change their circadian rhythm. And he played with them for 10 months. At the end of the exhibition, when I took him home, he followed me home one day and said, "Can me and my wife take them home? Because we want to take care of them." And they did. They took them home and they lived with them until they died. So it was like a really beautiful love story. And I was really interested in, sort of thinking through how research works and who are the invisible workers for us in terms of this pharmaceutical research that's done that we all partake in. We've all had aspirin at least, if nothing else. So I went to Symbiotica and did some more work there, which led me to further rats. And I got very interested in ultrasonic rat vocalization. Rats talk in an ultrasonic register to each other. They have they speak in a register that we can understand within our hearing range, but they also have a whole nother register far above what we cannot hear. And I was very interested in this. And I started working to try and adapt it for the lab. What could be better for the lab for these rats? Maybe not opening, plastic around them because it crinkles and it's really loud. Sounds like fireworks to them. So I've been working on this since 2010. And part of what I found then was this video and this research by this scientist, Jake Paxep, who was interested in, he's a neuroscientist who's deceased a couple years ago, but he was interested in ultrasonic rat research and communication, and he found that they giggle, that rats actually laugh. They make a kind of giggle register at 50 kilohertz roughly. And so here's a video of him. Whoop.
As we have listened to animals playing, we have heard what appeared to be the sounds of laughter. And we studied these for a couple of years without quite understanding that this might be laughter. And then one day we decided to tickle some animals. And we realized that we had to look at the sounds at a very different register than we can hear. So we obtained these transducers that are called bat detectors that can bring very high frequencies down to our auditory range. And when we did this and we listened in, we could tickle animals and generate a lot of vocal activity that appeared to be laughter. These animals would begin to enjoy our company, and they would start to play with our hands, and wherever we would put our hands, they would follow it. And when we tested these animals to ask whether they were enjoying this kind of activity, the unambiguous answer was yes.
So after 10 years of going to people's houses and going into labs and trying to register ultrasonic giggles, which I wasn't very successful at, I got 4 seconds over 10 years, I finally got in touch with Jake Paxep's colleague, Jeff Burdorf, and he sent me a whole bunch of files. So over the summer, we worked on trying to clean up those files and make a chorus of rat laughter. Like when you all laugh, it's a little bit contagious, right? Like if there's multiple laughing, I feel like laughing. I'd rather play this for them in the lab than have them hear country western music, which is what a lot of lab technicians leave on for the rats, thinking they like it. It's okay. So I'll just play you a little excerpt of the laughter. This is just one track. And this is downsampled so we can hear. So this is something we share with the rats, this ability to laugh.
When you say downsample, you mean just taking it to a hertz so we can register.
Exactly. Thank you. Yes. And this is the team that worked with me on this. So this is this research is ongoing at this point.
Quick question for you.
Yeah.
Has this sound been found in any other behavioral context or just from this
just from the tickling. It's a really interesting question though, because what we also found is there might be some other sounds going on. Like this is at about 30 to 80 hertz, so roughly 50. At about 22 kilohertz, was just above what we can hear. There's also another sound that we found in the recordings, which is a sound that they emit when they're fearful. So we don't know if they're fearful because they're being touched a lot, you know, and in these kind of containers like you saw, or what was going on. But these two things are registering together. So
didn't the scientists say that they thought to even perform the tickling experiment because they were hearing sounds?
Yeah, and you could see some evidence of their reaction to the hands and their tickling itself. So pretty much that's a happy register for them, and it's not some of the more anxious ones or fearful ones. So yes, in essence, yes. Yep. Okay, so let's. So I'm just going to go through a couple other projects. This is a project called Blood Wars. I've been interested in the immune system, as you could tell from the earlier rat project. In this project, I took blood samples from 2 different people and put them in a petri dish together to let the white blood cells fight each other. You see what happened. It's a little bit built on the World Cup tournament model. This design actually was done by Diane Bertolo, who's in the audience. Thank you, Diane. And so people would fight each other, and then whoever the winners were could fight, and then there'd be an ultimate winner. So oddly enough, people really wanted to give me their blood. I should have called it the Vampire Project, but and what we would do is I was trained by an immunologist to separate the white blood cells from the red blood cells, put them in a petri dish together after I'd stained them both with 2 different fluorescent colors. So one would be kind of reddish, orangish, and the other one would be green. And the thing that's amazing was that we had these, and then we put them in under an incubated microscope, so we could follow them with time-lapse microscopy for like 8 to 15 hours, as long as they lived. So it's weird, when you look at it, you have to sort of look like you're looking at a football match. You have to look at a certain part of the field to actually see their encounters. But they didn't always fight each other. Sometimes they made love. Sometimes they refused to do anything and just mutually died, like a kind of Romeo and Juliet thing. You know, it's like when I'm projecting all this stuff on it, the scientist I was working with hated these projections because he was an immunologist. And he's like, stop it. But anyway, I think it's really great because I also was trained as with these filmmakers who were called structuralist filmmakers, and this is probably as close as I'll ever get to making structuralist film. So. Okay, then we get into the gut work. I've been working with my own gut biome and gut biomes in general for the last few years. And there's something about poop, you know, now we're going to talk poop, okay. There's something about poop.
Get ready, here we go.
Yeah, I'm just giving you a warning if anybody's offended and wants to leave, you're welcome to, yes.
Hi, on your last slide, were those interactions real-time or real-time?
They were over about 8 hours, let's say, roughly, and they were about 1 frame per minute. So it's not really real time at all, it's time lapse. Yeah, I wish we could have done real time. We didn't have the hardware to do it. So we all know that, poop is a little shameful, right? So this is from this wonderful film of Miranda July's called Me, You, and Everyone We Know. And there's a scene in it where these 2 young boys are on a chat room, and they have this woman who comes in, and she's interested in talking to them. And they're posing as an older man, and the older brother turns to the younger brother and says, "What should I say?" And the younger brother says, "You poop into my butthole, I poop into your butthole, back and forth forever." And they sent it, and the woman said, "That's really hot." So there's something odd about poop. It's both hot, but it's also shameful. So these things are kind of overlapping as we think about it. I, of course, like many people here, probably have sent off my gut sample to be, tested by Ubiome. There's American Gut. There's lots of these places around the country now and internationally, and came back. And not surprising to me, because I have Crohn's disease, I have a kind of, altered gut biome from what a so-called normal person would have. And so that was interesting to me. There's lots of products on the market now for trying to work with bacteria. This is a bacteria soap called Mother Dirt, soap substitute, I should say. So we have a different kind of understanding about bacteria these days than we used to. I made a film a couple of years ago that was on fecal microbial transplantation. Because this is a technology that I became really interested in studying. This is actually I'll show you a video of it. I'm just going to show you a small excerpt of this, and this project was done, there's streaks on here, that's weird, was done in collaboration with Guy Schaefer, who is also a part of this. So it's just a 2-minute excerpt.
So FMT is fecal microbiota transplantation. And
Thank you.
what we do is we take a donor stool and we transplant that stool into the recipient's body. The whole goal is to, in essence, we are transplanting stool, but the goal is to give the new microorganisms, the bacteria, and to have a chance for the new microorganisms to actually implant into the recipient's body. And we do that either through a nasogastric tube, where we make a solution of the stool. We do a colonoscopy and then take the donor stool's solution and spray it into the recipient's intestines. We can also offer it through enemas or through flexible sigmoidoscopy, which is like a mini version of colonoscopy.
OpenBiome actually started part out of some of the research that I was doing at MIT at the time, investigating the human microbiome, and then in part also out of a family friend who ended up getting Clostridium difficile infection. He ended up going on 7 rounds of vancomycin, which is sort of the most powerful antibiotic we have for C. diff. So failed every antibiotic treatment that he was on, and was very, very desperate for an alternative, something that would work for him. He ended up doing fecal transplant at home in his apartment with his roommate's stool, and it worked for him.
So that gives you a little window into what this technology is, and I researched by interviewing gastroenterologists, scientists who are working with synthetic gut material, in other words, kind of taking the bacteria out and putting it back together in different ways. And so it's just sort of looking at the state of the science. And it is a paradigm shift as to how we're going to be treating people in the future. I mean, not just with poop, but how we think about our gut microbiome, what it can do, and the potentials of it. So someone had asked me if I could have a FMT with anybody, who would I pick? And of course I thought, why not ask David Bowie? He was the first person who came into my mind, and this was before he died. And I decided I would make a series of photographs imitating his work that was the most interesting to me, and send them to him, and ask if he would exchange them for these photos, his poop sample for these photos. I have a bargain for you. I'm writing as a strange request. I'm a lifelong fan. I've been following your career since I was little. I'm a little bit younger than you, like a little sister. I offer these photos to you, reenacting famous images of your career, et cetera, et cetera. Tilda did this before me better, but I'll humbly offer mine among the others. I was hoping these photos might capture your attention for a moment. I want to exchange these for a throwaway item, your poo. I want to conduct a fecal transplantation with your stool, implanting your poop in my gut biome. So what I thought really would happen would be that I would become a little bit of Bowie. And who wouldn't want to be that? So I feel like it's almost like an organ transplant, and that is actually how it's treated by the FDA. It's not like blood sampling. It's really highly regulated. So there is something very different in terms of how legally, even within the medical institution, how poop is being regarded. So maybe that was right. Now I sent this to David Bowie just the year before he died. He was very busy, and I'm sure he had lots of things on his mind. So I didn't hear from him. So I was lucky enough to work with a scientist, Will DiPaolo, who was working with the gut biome, and I've been a resident in his lab for the past few years. And this is Will and myself working with our own poop samples. We decided to test them side by side, me being the sick person, him being the healthy person. Bless you. his lab had, interestingly enough, not really worked with kind of, like I want to call them live poop samples. So he'd gotten poop samples from people from hospitals that were anonymous, but not anybody's they could identify. So the fact that I was working with their boss's poop made people in the lab giggle. And we designed this protocol where, and Will did this, where we would take blood from each other and use the white blood cells from the blood with the fecal matter to test to see if there was any reaction from the white blood cells to the fecal matter itself. He had a theory that this would happen with me because of a phenomenon called leaky gut, where in my gut, because of my Crohn's, probably the stool had passed into my blood system before, so it probably was recognized as that. And so, you know, we did this, and again, we're testing it over time. His stool sample and my white blood cells and his white blood cells is pretty unspectacular. And then we tested it, my poop samples with the white blood cells, and this is my material on the top, me against me with me, I should say. And this image was really startling to me because it's an image of T cells reacting to the material, the poop sample that's coming in. The T cells are going like poof, and it looks like an explosion. And at first I was kind of horrified. You've always heard as a person with immune system issues about T cells, you know, T cells going crazy or your overreactive immune system, and it's hard to try and imagine what that is. This gave me a kind of imaginary of it. This visual helped me understand that. But it also made me think maybe I'm not just like a sick person, which nobody wants to think of themselves as that. Maybe I'm some kind of mutant preparing for the future. Maybe this is like the way things will go in the future. Maybe this is actually ahead of the curve. I don't know. So I've designed a project called Family Biocrests, where we're taking samples from people who live together and seeing what they share. And we've got lots of material on this. And here are some of the biocrests. We're making these in 3D now. I'm working with an artist in Philadelphia. And these are samples that people come up with together. So this is a household of 3 adult females, 1 infant male, and 1 female cat. And these are the bacteria that they share. And there are lots of other bacteria in there, but these are things that cross. Here's another example. When I was doing these plates with Will, one of the things I began to notice was that there were areas of my plates that were empty over and over and over again, repeatedly. So we would have these plates with his bacteria nicely growing in this agar expressing different kinds of bacteria, and my side would be empty. And this kept happening over and over again. And so one of the things that this means is that I have a kind of dysbiosis, meaning I don't have a real kind of diversity of bacteria in my gut because of the antibiotics I've had, et cetera, et cetera. So we all want to think of our guts as a hackable space. Sorry, when I creep ahead, it's terrible. And I'm trying to think of ways to work with that. Okay, if I have dysbiotic, what does that mean? So my friend Guy Schaefer and I, again, made another video called Okay Poop It, and it's actually a video that's sort of like a promo for an app that we're developing. And it's a kind of counter to the other video you saw because it's more DIY. We were interested in thinking about this if people could have this technology for themselves, as opposed to having it be so heavily regulated. There are problems with that, so this is kind of a spoof. But I'll just show you a little bit of a sampling from that. Here's the results of one encounter, and I'll show you what happens when we have our first Okay Poop It date.
Oh, hi.
Hi. So good to meet you.
Really good to meet you. Thank you so much.
Of course. Yeah, I really want to be able to.
And this is actually my collaborator.
Really, really great. That's awesome. You're the first, so I'm a little nervous.
So, thank you. I'm really honored.
Yeah, me too.
Yeah.
No,
Oh, of course. Yeah.
like, and I wanted you to be first, so, you know.
Really touching. I mean, it means a lot to me that you thought that. So thank you.
Yeah, so maybe we should, I mean, maybe we should just get down to business and I'll ask you some questions.
Yeah. And feel free to ask questions.
Oh.
I'm an open book with regards to my poop.
Oh, thanks.
You're welcome.
So, like, what do you like to eat? I mean, are you vegan?
Oh, not at all. I eat everything. I mean, I'm a fairly, there's a lot of salad, a lot of oatmeal, a lot of whole grains, a lot of, roasted vegetables.
Mm-hmm.
But there's meat. Any meat. Yeah.
Meat, okay.
Fish, yeah.
You meat, okay, yeah. And how's that work out for you? I mean,
the meat, I'm a little sensitive with regards to meat.
Yeah.
I had kind of a big pile of meat last night, and it didn't, there were problems, I mean, like, so, sometimes I overload the meat.
Oh, okay.
Yeah, sometimes on a backside, it hurts, and yeah.
What sign are you?
I'm a Scorpio.
Oh, me too.
Oh my god. That's so funny.
We might really be compatible. That's
Well,
amazing.
yeah. Of course, Scorpio poops. So
Yeah.
That makes sense.
So amazing.
So, I think that because of timing, I'm gonna stop there. And if anybody has any questions, I can answer them, I guess, now. We're going to
Yeah,
the
so
next.
let's actually bring Alison up, and we'll kind of answer questions all together here. So maybe you guys have questions. I have some questions, and just by way of providing kind of an inflection to this question period, let me just say a couple things about what this proposition of bringing scientists and artists together and making two presentations is for me. What my interest in it is. So, my name is Ed. I work with Susanna over here. And you are sitting in our studio space, which is the Canary Lab. And we produce art, media, and programming about ecological issues here on campus. And I'm telling you this because, for me, the interest in both art and science and in bringing them together is in thinking about what ecology is. And for me, ecology is about relations and revealing those relations and seeing the world as a world of relations rather than a bunch of silos, and including relations between artists and scientists, potentially. And for me, too, the art and science are both two different epistemologies. That is to say, the tendency when thinking about art and science in juxtaposition to each other is sometimes to resort to certain stereotypes. Art is a decorative activity. It's about the fancy colors and lines and stuff like that. And there's equal prejudice we might bring to the science side of it. But I think of them as two different epistemologies, two different ways of knowing or two different ways of revealing things. So I just wanted to state that by way of framing some of the mutualities you might discover in this questioning period. I have a question, I guess, for both of you. And I'm happy to open it up at any time. Like, I can ask questions, but I'm happy, if you guys have questions, just jump right in. But I'll just start with one. So Kathy, I noticed when you talked, you're like, well, I'm not sure how I'm going to resonate with Alison's talk. I work with bacteria a little bit. But one thing I noticed that was a strong presence in both of your presentations was the word imagination. And you actually, an inspiration. And you actually at one point said, this is what really inspires me. And I was surprised by that. And so I wondered if you would talk about the role of inspiration in your work and imagination in your work. I guess those are two slightly different concepts, but link to nothing. So what about inspiration?
Yeah, I wasn't prepared for that one. I mean, come on, what's going to get you up every day and keep doing what you're doing?
Right.
If I wasn't inspired by this work, I probably wouldn't be doing it. But, as you probably saw, I do a lot of image analysis, a lot of movies, and microscopy, and that really, I'm really hands-on in the work that I do. Mechanical engineer by training. so I like that tactile sense, and I can see, and the fact that, you know, I get a beautiful result at the end of the day is really nice.
What do you think inspires that? Like, what do you respond to? So you
It's
both-
learning
yeah.
something about the world.
Uh-huh.
Yeah.
And what about for you? What about imagination for you, or
Well,
inspiration?
I mean, I really like that, learning something about the world, because I think for me, I'm just curious about a lot of things, and that's what takes me into the lab. In terms of imagination, I mean, when I was working with Will DiPaolo in his lab, he was really great because he really felt that the art actually informed his science as well as the art helping, you know, going only one direction. Like scientists sort of being taken advantage of by artists. And I think he felt that when I was in the lab with everybody there, he's a great team, that I would be asking these kind of macro questions, I guess, or, you know, they were sort of like stepping out of, scientists have the task of having to look at very sort of focused, you know, questions for a while. And then they can go to something else, but for a while they're kind of locked into something like that, I found, in his lab. And so I would come in and I'd kind of blow it open a little bit. And so we could go between these modes, and it felt really great. And I guess that was part of the imagination part.
Does that resonate with you? Like, you asked two questions at the end of your presentation. One was what I would describe as a technical question about can we assess something about the flows, and the other one was more of almost a metaphysical question, or a question you normally associate with the humanities, in terms of what does this say about what living material is, right? Is that second question one that you would answer in the normal flow of your practice, or what was your intent in proposing it?
So I like that spin. That would be great if we could contribute to that aspect. I guess I was thinking still on the technical side that we can, but I think we have to make new physics to understand these materials that isn't there, and we don't know what that quite looks like yet.
Have you ever collaborated with an artist before?
No, I have not.
One of the things we like in our fantasy about this bioart mixer is that it would spawn collaboration. If you could collaborate with an artist, do you have any sense of how you would do that?
I mean, I have lots of movies and images, and I hope I could learn some things. So I'm always trying to represent schematics, or when you're just making a figure, it's really a story. You're trying to convey something, and I think I could learn a lot from artists how to do that.
I have another question for you.
Yeah.
It struck me in looking at some of your work that I might place it in a category that may or may not be dubious, or you may or not have a positive reaction to, which is the aesthetics of care. That it produces a sense of care or a feeling of care.
Yeah.
And is that intentional, or is that a sort of yucky association for you? Or is that a good association?
No, I think that particularly for example, the embracing animal piece for sure was in that area, that realm of the aesthetics of care, meaning, you know, it's looking at how we care for others. So yeah, no, I think it would fit into that. I think one of the things that I find really interesting in bioart, and the artists that I particularly am interested in, is that those artists are, and myself included, are very much asking ethical questions all the time, and looking at ethics around the cells we're working with, the animals we're working with, you know, and how does the killing happen? What is that like? And bringing attention to it in a way that, like the Tissue Culture and Art Group have a really interesting workshop where they develop things using different tissue culturing, right? They help ask their participants in the workshop to develop sculptures or something. And at the end of the workshop, they pass the plates around. Everybody opens them up and touches them. In other words, you know, the cells are going to be killed by have bleach put on them or put down the drain, or however people do it. But this is a way of accelerating that process by introducing our own bacteria into it. And they want people to be conscious of this kind of, you know, activity. And I know people who are in the lab I'm not saying that scientists aren't conscious of this, because most of the time the scientists I work with are very conscious of it. But I think that it's hard, as you know, as we go through this sort of accelerated life, to always think of those things.
Yeah, I mean, whenever you think about care, it's how far you're going to extend that sphere of care to what species, to what end, to how much sentience.
Yeah.
And one of the things that I was struck by in your talk is that to me, it raised questions about intentionality of the individual bacteria, right? So and I'm curious to what extent your research has revealed like levels of intentionality of movement. They're heading towards something, away from something. There is a reaction which you can say that's automatic. But do you know the biologist Jacob von Uxel? I think he's more popular in the humanities than the sciences at this point. But he has this concept of umwells, and he talks about a tick, and that a tick only senses 3 things in all the tick's life: light, heat, and the sense of butyric acid. And in the absence of those 3 sensors, the tick doesn't move at all. It's frozen in time. And von Uxel put a tick in a bell jar, and the tick didn't live for 18 years. It had nothing going on. It's barely alive. And so bacteria is one level even simpler than that. But in some senses, does your experiments, to what extent, I guess is the question, does your experiment reveal some sense of intentionality in the bacteria?
Yeah, that's a good question, because I often, for creative purposes, do try and imagine, okay, I'm a cell, what do I want to do?
Yeah.
But then I have to ultimately I take a step back and say, okay.
I'm going crazy.
Well, yeah, we can't describe the cell. Yeah, because it doesn't have wants or needs or whatnot. But, I mean, it's interesting about the tick.
Mm-hmm.
I'm surprised it's so simple, because I don't think bacteria are that simple. They respond to way more things than just 3 things. And you can ask the question, why do they swim? And you can figure out that, okay, well, if they're just going to beat out diffusion to get to a nutrient source, that's going to tell you just how much they have to be able to power themselves. And is it going to be enough to require all that energy to develop an apparatus to even move?
So once you establish that there's something towards which they're drawn or from which they're repelled, then they have preferences. Or is that going too far? Like, at what level does it become a projection? Like, I noticed someone asked a question to you about the laughing, and I felt like maybe, were you trying to say, are you sure that sound is laughing because you can only identify it from the one instance in which they're being tickled? That's how I interpret it.
Yeah, and to see if it occurred in any other behavior, so if it could be seen as laughter or if it could actually be communicating something else.
Mm-hmm. That's particular to this, like, this hand is coming and interfering with your space, potentially. I mean, how sure are you that it's laughing?
Well, I mean, I haven't really been able to work directly with the rats while that sound is being produced. As I said, I got 4 seconds over 10 years. From the research I've done with the scientists who have been doing it, they are able to observe this and hear this register when rats are playing together themselves, and mostly it's like younger rats, like kids playing and wrestling and having fun. And so that's the only register they get at that point, is when what they're observing looks like it's kind of fun stuff, and then they're hearing this emission from the rats. I think we don't know a lot. I mean, I think that one of the things I'm interested in thinking about this kind of level of ultrasonic communication with rats, but we're going to be going into other animals too, is it's dense, and I think we know this much about it. And I think that's a really good question, and those are the kinds of questions they're just beginning to surface, because the technology is just beginning to catch up with what we can hear. So you're spot on in that we have to keep asking these questions.
Yeah, let's take some questions over here. Yeah.
I feel like that's like a common disparity between science and art, in that I think scientists, like, what you're sort of scared of is like bias, and like how is your anthropogenic thoughts going to affect what you're doing with your research.
Yeah.
And so I think like a lot of scientists are scared of trying to, for instance, in the rat case, be like, oh, this is laughter, we want to try to break it down into things that we can understand.
Right.
And so, I mean, in the instances that you were talking about, like with the films that you've made, it was very helpful to bring science into the humanities, because like humans understand things through art, and it's hard for them to look at, if they're not a scientist, in that So I think like it's really hard striking that balance between art and science because sometimes, you know, if you tell a scientist that's laughter of the rest, they're going to tell you, oh, it's probably not, because they don't want to change like the research.
No, I've had that exact conversation, in fact, with some scientists who say, well, it could be an expression of joy, but I don't know if I'd call it laughter. So you're exactly right.
And there was another moment in your presentation where you said, I'm quite willing to make these projections, making love and mutually dying like Romeo and Juliet and so on.
Yeah, but the scientist I was working with was devastated when I kept saying these things. He was like, stop it. They're just exchanging fluids. I'm like, okay.
Yeah.
Oh, I have a question for Alice.
Sure.
So like, I noticed that when you like showed us like the vector videos for the bacterial movement, it was kind of like on a plane, right?
Yes.
Like, is there ever like a case where it's more like in a field, like a space, you know, like a 3D space instead of like just flat? Because like, I don't know, like I'm not like quite, I don't know much about it, so I was just wondering like it would be like more accurate or like there's like more to see if it was like projected in like a 3D space instead of just like a plane.
Yeah, that's a good question. So the swarms I showed you are pretty much a monolayer thick, so we like to think of them as basically 2D. But there are bacteria that can form 3D structures, fruiting bodies, and or a biofilm is usually thicker, or they can swim in 3 dimensions, but usually not in such a collective way. So there are ways to do that 3D analysis, but it's quite a bit more complicated.
I was really intrigued with the last slide that you put up with it had the red lines that were tracing the kind of patterns of the behavior and immediately wanted to incentivize the behavior by adding nutrients to certain areas of that slide or changing the temperature and viscosity of the material. And it struck me when you were talking about somewhat in the backdrop of the rules of physics in terms of observation of materials. And with living materials, I really think you're almost forced into being in a relationship. So usually when people are just observing things, they're really concerned with over-influencing just in the act of observance. But it seems like if you're going to push physics or engineering into a living material aspect, you're going to have stronger relationships with the material you're studying, and that seems to be possibly an impediment in terms of your own discipline in science.
Yeah, so that's an interesting question, and I kind of want to tell a story about when we first discovered microscopic motion, when people first looked at like pollen grains and under a microscope, and they saw these things moving around. And they said, oh, they must be living. What are these small organisms? And actually it took Robert Brown, multiple tries to convince people that, no, these things are dead. This is a property of dead material. And that's Brownian motion. So, and there was kind of the inverse bias, right? So I very much like to look at that interplay of things that are subject to all these physical forces, fluid flow, Brownian motion, and yet because they're living, they can explore different spaces and do new things that dead material can't. So yeah, I'm very aware of that line.
I was really interested by this contrast between the work that you're doing in that these ideas of collective motion across scales of life, and that we're even comfortable saying that a similar thing is happening with mosh pits. So when it comes to like physics and the physics of interaction, we're fine saying the same thing applies from humans to birds to bacteria to non-living things. But when it comes to something with emotions, we're not comfortable making those, or scientists rather are not comfortable in the same way of making those connections across living and non-living things. And that difference when it comes to behavior and relation and physical property.
There may be a good reason for that distinction, actually. I think that's an open question for me, is to what level those need to be broken down. And that's, when I think of these two different epistemologies, I think of them in collaboration with each other, not in some kind of fight, And so you were saying, to answer this question about the living material, we need a new physics. can you imagine that this exists, or maybe you just need the humanities? Anyway, yeah.
No, I think these kinds of teams are important now, in terms of, you know, having various people from different disciplines come together to work to figure out these kinds of problems, because I think each team member is going to add something new to the formula, which can override our own biases, which are evident in all of our practices. So, you know.
Yeah.
Alison, in the preliminary analysis of the mammalian cells, are you seeing similar vector patterns from the bacteria to the mammalian cells? Are the somatics like overlapping in similar ways, and like the way the policies are working?
Yeah, that's a good question. So, I guess I just wanted to demonstrate that there's collective motion in the sense that you have many cells moving in the same direction, and that's about it.
Okay.
We haven't been able to compare more of the details of the cortical flow structures yet.
I have to ask Kathy about the thinking you've done on the biome exchange and the fecal transplants and things like that. You spoke about the regulation that was there and was probably necessary, because we can imagine a world where there's a vernacular use of this activity.
Yeah.
What I would assume that would happen would be that there could be a kind of crushing of difference between biomes, and there could be a global biome, or at least speaking of a community.
Yeah.
And I think that's interesting, because as I understand it, that the biome could be used to reflect individual identity, and so that could cause a kind of a community problem.
Yeah, I think that you're right. I mean, I think ultimately we'll someday graduate to, you know, like possibly gut biome identification as opposed to fingerprinting or that kind of thing. Much like we're relying on DNA now. But it's interesting you're saying that, because now there's real interest in groups of people who have lived very isolated lives, like on islands or things like that, and had a very simple diet. And so there's, you know, like this kind of poop piracy that's happening, where people are actually going to these different places to try and collect these samples so that they can try and, you know, either deconstruct them or look at them, because of this is what's going to happen, as you said, is that things are going to start merging.
Colonization of poop.
There is. It's really an interesting time we're in, I mean, I think, in terms of this research around the microbiome.
Yeah.
I'm a molecular biology student here, and I'm in a microbiology lab right now. And I feel like it's been a little bit difficult for me to like stay within the science field. So I'm really excited that this was happening, because I feel like all of my classes are kind of this like breeding ground for human supremacy. And it's like we always are like looking at these things that we're gauging intelligence on, like the ability for someone to use tools. And I'm sitting in my microbiology class and I'm like, oh my gosh, all of these bacteria are literally using us. You know, they're getting us.
Right.
We are their tool. They're getting us to sneeze. And not only are they getting us to sneeze, they're getting us to kind of get satisfaction out of it. So I was really appreciative when we were talking about kind of that like pushback of like the Romeo and Juliet of all of the microbes and stuff. And I kind of am just glad that it's like a wider discussion, because it's been so difficult to kind of have those conversations.
Yeah. No, I appreciate you saying that, because I think that this thinking about symbiotic systems that we are all living in now is sort of, I think, one of the keys that we have to trying to tackle this climate crisis we're in, if you will give me that, because I think ultimately if people can start understanding these kinds of relationships, and then we're not alone at all, you know, then maybe we'll start waking up a little bit.
I'm glad you mentioned the climate crisis. I try to remember to mention that every time I do a public event, because it also, I mean, it demands these types of interactions you're talking about in these kind of collaborative teams. Yeah.
Kathy, can you talk a little bit about C. diff and what it is?
Oh, thank you. I'm sorry if that wasn't clear. C. difficile is a kind of, it's a bacteria that we all have. We all have this in our gut. But it can, if it overgrows, it can really, well, ultimately can kill you. And so patients that get infected with C. diff often are patients who are ill and in the hospital. And so their immune system is already kind of weakened, and then it's an opportunistic moment for the C. diff to sort of propagate. And when that happens, the problem is that scientists have found, I mean, gastroenterologists, medical people, and scientists have found that the antibiotic treatments that we have don't work. So we actually, this film goes on, and we actually were able to get into a fecal transplant in the operating room. And the patients had given us their approval, and they were really happy to get this word out because they had gone through like a year of just constant diarrhea. And they were like, just tell people this is like the FMTs, the fecal microbial transplantations, have a 90 to 95% success rate in healing C. diff. So it is kind of in that world of C. diff, a kind of miracle drug for people at this point in time. And there's obviously problems with it. There's been cases where, there was a case where, a couple cases I think, but one case where somebody died, they were infirmed. There was a case where somebody became obese from the fecal matter they had received. So there are lots of questions around it still. But I think it's really, really, there's positive research in it. And if somebody's has an option of like maybe dying or taking a fecal transplant, they want that option. The other thing is it's the only place that it can be used right now. There are trials that are going on for other diseases, but C. diff is the only thing that we can use for FMTs for in this country. Yeah, thanks.
Any questions?
Just generally speaking, we're talking about bacteria from both areas, and as a living substance, but viral bodies have a different motility, and they have, they move because of replication, et cetera, et cetera. Could there be a closer correspondence to material understanding with viruses rather than bacteria? And could there be biomes or viromes?
Yeah, there is. Yeah, there is. Actually, the next research I'm going into is with viruses in a lab in Philadelphia. They work exclusively with viruses, and I'm really interested in macrophages. And I don't know very much about this. I'm extremely excited about that research. But I think there are virus biomes. If that's what you call it. What do you call it? Viromes. Thank you.
Yeah. But is there an understanding of viral motility that could be?
I don't know if they self-propel in the same way I've been using the word motility. So there's a different language for that.
Okay. Well, there's a third phase to this, which is yes, it's conviviality time. So to make space for that conviviality, as you get up, if you can start stacking the seats and we can push them to the side.
I just want to make an announcement about our next BioArt mixer, which will be on March 27th